as a consortial activity between us and University of Alaska, New Mexico, primarily. Involves a tremendous amount of staff time from all places, but particularly Carla and Michelle and Chris. Also, the data in Arctos, and whether it's the scanned images or the specimen data, comes from you all. So our data is built from the activities of the MVZ. Sort of filtered through, mediated through a wonderful set of undergrads who've been working with us for many years, people like Kura, which is here, been doing a lot of the scanning work. So I just want to acknowledge the effort and involvement of those undergrads. The other point to make is that the data that goes into Arctos obviously comes from us, and it behooves us to try and get our data in the right form, the right quality, to make the curatorial process as simple as possible, as efficient as possible. We'll have more to say about that as the semester goes on. But for now, what are the cool things you can do with Arctos?
Okay, thanks, Craig. It is true that the quality of the data we get is important, but we're not going to talk about that today. So there was an opening in the MVZ lunch seminar, so we thought we'd take this opportunity to demo Arctos for new people and also show people that have been here for a while, for a long time, some of the new and cool things that you can do that we're working on in the development. So here's the link right there to actually search the database. And we have a website that we're working on, which is arctosdb.wordpress.com, so that has more information and documentation and so on. So what is Arctos? So Arctos is an ongoing, and I emphasize ongoing because it is continually under development. I'll get back to that a little bit at the end. Ongoing effort to integrate museum collection data, mostly natural history collection data, collection management tools, and external resources on the web. And I'm not going to talk so much about the collection management tools, like how to, loans and accessions and so on. That's all integral to Arctos. We really want to focus more on sort of the research, what you can do to maximize access to multiple data types for biodiversity research. I'm just going to give a brief overview of Arctos, and then I'm going to leave it to Carla and Michelle, who are actually going to be doing demos and showing different features, but we want this to be dynamic, so if anybody has any questions as we go along, feel free to interject and ask them. So, as Craig mentioned, this is a collaborative effort. There's 2 instances of Arctos right now. One includes 7 institutions, here's MVZ, mostly in the west, up to Alaska, and then including Denver. 39 different collections, and 1.4 million specimens. And then there's a separate instance right now at Harvard called MCZ Base, so that's also based on Arctos. 7 collections, 1.6 million specimens, and there's been a lot of discussion, and we're still discussing merging these 2 into a single merged instance. It's important to also emphasize that Arctos is part of the broader data network community. Most of you are probably familiar with ORNIS, MANIS, HERPNet, FishNet, maybe not so much, but FishNet as well. These 4 distributed data networks are now being merged under a single project called VertNet. These are all NSF funded. VertNet's an active project. We just got funding in May to do that. And right now there's 72 institutions, I believe, and 74 institutions, and something like 84 million records in VertNet. Of the 84 million records, that's over 60 million are actually observational records from the Avian Knowledge Network, which is like eBird and Project Feeder Watch and things like that out of Cornell. And there's another, how many collections on the waiting list, or institutions?
64.
64 institutions that are on our waiting list, which will be part of VertNet, hopefully within the next year or so. So I want to emphasize, I think one of the unique things about Arctos is the diverse kinds of data that is included. So specimens are obviously at the core of the database system, but we also have audio, and we're going to be getting a lot more audio within the next few months. We've got all of MVZ's audio recordings digitized, and I'm just working to get those linked. DNA sequence data through linkages to GenBank, relationships between different kinds of data, so parasite-host relationships or predator-prey relationships, lots of GIS data, all of the data in Arctos, essentially all of MVZ's data, and most of the data in Arctos are georeferenced. We use projects as a way to sort of group different kinds of accessions or different kinds of research projects, and we link specimens to publications. So it's really important actually when you guys publish a paper to cite MVZ specimen numbers, because we link the publication directly to the specimen, and that's how we can show how valuable our specimens are. And then field notes, we're working to integrate our field notes into Arctos. We have some field notes in there right now, annotated maps of collecting trips, and images both of specimens and also habitat and landscape shots. So people that are doing field work, if you're taking images either of your specimens or the collecting sites, we want those as well. I mentioned different collections. There's lots of different collection types. So obviously MVZ is mammals, birds, and herbs, as well as eggs and nests. There's fossil data, so paleontology data from Alaska, entomology data, fishes, and plants. Mostly those are from the University of Alaska herbaria, fish collection, and insect collection, and paleo collection. Arctos is also unusual because we include observational data. So if you are hiking somewhere and you see some unique snake and you have a camera but you don't have a permit, you can take a picture and we'll actually catalog that into Arctos. All of the bird observations that Morgan took over the last few years as part of the Grinnell Resurvey project, we're going to put into Arctos. So those will all be, so you'll be able to query not only the specimens that were collected, but also the bird observations and all the images from those point count transits.
We also mentioned Andrew Rush contributed.
Andrew Rush as well, right, for Yosemite, exactly, and a host of undergrads. So this is just a sort of a schematic of Arctos. This is focused on MVZ Arctos, but you can apply to the broader Arctos. So some of the basic functions are, allowing you to search, enter, and update data, georeference and map localities. So there's visualization tools and also georeferencing and mapping tools that are directly integrated into Arctos. And I'll show you that in a minute. Manage transactions like projects, loans, and accessions. Track specimen citations through publications in GenBank. Track collection objects, so like tissue. So we're spending a lot of time right now doing tissue barcoding all of our tissue vials and putting those into Arctos. And then there's a lot of linkages to external web resources. So all of our field notes, images, sound files are being archived at the Texas Advanced Computing Center and then directly linked to the specimens. We also have images that are archived in CalPhotos that are linked. Right now it's not dynamic, but we're trying to get money to do that. We've got some linkages to Digimorph, which I'll show you in a minute. And so with all of this sort of multiplex of data, it's very powerful in terms of asking biodiversity research questions. So Arctos is used by, obviously, people like us, academics, museum curators and researchers, but also a host of other kinds of people, federal and state agencies, and they also contribute to Arctos, contribute specimens to Arctos, non-government organizations, consultants, teachers, and also artists.
Carla, can I just ask a question
Sure.
of the audience? So who here among the students in particular has used Arctos? And who has not used it or liked it? There you go. Just curious.
So just want to end on this. So Arctos development is guided by you guys. So people that are using Arctos, if there's features that you think are missing or that aren't quite working the way you think they should be, you should tell us, because that's how we guide development, is by getting feedback from researchers and also curators and data managers. So with that, here's the interface, and I'm going to turn it over to these guys to actually do some demos.
All right, switching seats here. I'm going to grab this. All right, so as any of you guys may know, when you're standing in front of a large audience and you want to do something as ambitious as a live demo, there's a certain amount of prayer needs to happen. So we're just gonna wing it here. Okay, so we're live on the website right now. So one of the nice things about Arctos, we can almost do all our curation, and you guys can do all your searching from any browser. So it's always live, it's always on, and it's always available. So here we are on the internet. You can also find links to Arctos' search page from the MVZ homepage, and we have it sprinkled in other parts on our MVZ website. So when you first entered, I'm just going to actually also use my cheat sheet because we have a sort of a script here that I wanted to make sure that I introduce folks to the various parts, especially for those who are brand new to Arctos. When you first enter on, we're using this as a public, non-registered, non-curatorial user, but you can see that you've got already a lot of options here to search. This is the most basic search boxes here, and if Carol can open up some of these links right here, you can see that you can actually drill down into much more complicated, or much more, I should say, not complicated, but much more specific searches if you wanted to. So you can actually search down, you open up this drop down right here, Carol. You can see that you can actually search on things like GenBank numbers, or there we go, or collector numbers, which I think is farther down in our list. That's all right. If you go to our collection drop-down list right here, you can see that we actually, when we're not registered as an MVZ user, you can actually have access to the entire Arctos community right here. And we're going to show you actually some of the power of having multi-institutions within one collection database. So here we have the various acronyms of the different institutions and their collections. We also have, okay, thanks.
Sorry.
Yeah, that's right.
Keep going.
You can close that up now to show a few more options. And our locality searches, which is also very popular. Some of us are a little bit more visual, so you can actually create bounding boxes right within a Google Maps interface to grab the bounding box coordinates and do a spatial query. You can go ahead and find that. Okay, and then lastly, I'll just show you a few more things. Maybe you can just show us the options of your data collector. For those who are actually contributing to the database a lot, oftentimes you want to just see what's been cataloged, what's available. Where was I in 1975, Ted? So you can immediately kind of go in there. I would suggest using your last name because we often use people's full name. So sometimes if you're not used to doing that, you might put in Dave Wake and it won't return. You won't have a satisfactory return. So if you put in just the last name, you should be able to find that. And then, and as you can see, there's other parameters you can add in there. So once you go and collapse those, and we're just going to do a quick search on and see what's MVZ's first perf specimen. And we'll do a search here. User. And so the first thing you get is the specimen results page. And if we had a larger search parameter, we would get actually more results here. And this is sort of the default view. Actually, we can get specimen results as in a table format, but in this table format, we have links to a variety of other things. So I'll just quickly point these out. If you click on this link, all these blue letters, I should mention, anything written in blue is a hyperlink to some other attribute or some other table in Arctos. So here we could do a search actually on all the specimens found at that particular specific locality. Likewise, we could also do a search on all the specimens with this taxonomic identification. We're just going to also show you a customized form right here. Because sometimes you actually want to see a little bit more detail in your specimen results. So we can actually see the various attributes for that particular specimen search, or locality information, or various other things. So we're going to actually close and with that refresh, so we don't have to see that. Yep.
So can you just quickly show how you would search for tissues?
Oh, sure. Why don't we? I'll show that when we do one more search, because actually I think that's later on. We do a piece of search, but yeah. That's a good question. You can
go back to the front for a second.
Sure. Let me go ahead.
You go here.
Yeah. So required tissues. just check that off, and then we would probably not specify a specific specimen number, obviously, and but let's say we did a taxonomic search, so then you would get all specimens of that particular taxonomy that has tissues. Likewise, you can also require media types, images, sounds, or maybe you want to see everything, including observations or only observations. You can do that as well. So, okay, so let's go back to our results page. That's the last thing I wanted to point out. Well, and then, of course, if you click on the catalog number, that will take you directly to the specimen detail page for that particular catalog animal. The specimen detail page has a sort of a, predictable layout. So I'm going to briefly go through this, and you'll start seeing this over and over again as we go through other examples of things. But you have basic information summarized up here at the top. We have the taxonomic identification history over here, so we can see that its first identification was under a previous name, Crotophyton ciliaris, and now here's its new identification. We have this whole block of text here will have everything to do with its locality and georeferencing. So we can see the name, the locality that as it is being georeferenced, and here is some of the gory details for the coordinates. So it does have coordinates to it, as well as a collection date. Yes, exactly. Yeah. So this is done under HerpNet, and the tiny print, you can actually see some of the georeferencing sources that was used in any specific remarks. Collectors, we also have other identifiers here. I'll be pointing this box out to you later on. Some parts, some information about that particular specimen. The accession number is more of a curatorial function, so most people aren't that interested. But I'll return to this project later on. But here you can see that this particular specimen is actually also included in one of our historic Grinnell surveys. So up at the top, I'll just point out one more thing, I think that's also on my list, is some of the mapping functions. So we do actually have a little thumbnail here. So there's two ways of mapping. You can also just dynamically map through Google Maps, or you can toss it over to Berkeley Mapper, which is our mapping visualization plugin tool, which gives you a little bit more interface tools and better base maps than Google Maps. For some of our specimens, you'll notice that there's a second Berkeley Mapper button that says Berkeley Mapper plus range maps. And we're in the middle of actually trying to upgrade that, so we'll have the most up-to-date IUCN range maps. So you can overlay points on top of range maps, expert opinion range maps, which helps, again, put sort of the specimen results in context of what the range of that particular species is. Okay, so let's.
So Michelle, can you do multiple range maps? So if you've got 2 or 3 species and you want to see where they overlap, do you have that power?
No, at the moment we don't. Yeah, and at the moment, actually, I should also mention that we're restricted to the MVZ's sort of taxonomic range. So North American birds and mammals, global amphibians and reptiles, and in part that's just has to do with the availability of the IUCN range maps right now. But actually we're talking to IUCN, and they are developing web services, and we're trying to incorporate those. And so as they, increase their range map assessments, we likewise should be able to see that. But that is something that is actually we've talked about including. There's all sorts of complications.
I know.
Yeah. Okay, let's go back to the search page again. We're going to do our MSD here. So we're going to show you, by example, some of the, like I said, the benefits of having multi-institutions included here. Here's, again, specimen results. We're going to go to the specimen detail page. And you can see quickly, this is MSV parasites. So they've got actually I think it's NSF, right, to catalog parasites in their collection. And at the bottom here, unlike the other detail page, there's a relationship box. And that relationship box points to relationship of this specimen being a parasite of UAM mammal 78448. So let's take a look at that. And now we go straight to the UAM collection within the same Arctos interface. So if you go down to the bottom, it likewise links back to that MSB parasite. Having that multi-collection involvement, allows us these sort of cross-collection talks, which is really important, as you can imagine, if you're trying to do these kind of complex ecological studies, or even trying to cross-validate, various food webs or what have you. Let's see. I'll also point out one more thing here about citations here. I know.
I love the parentless voting.
Yeah, I know. You're not supposed to say that. Sorry, we lingered too long on that. Okay, so we'll go back to the search button at the top here. Let's go through the search button here. On the search button, so we just showed you the sort of the default first page, but we actually can search on many other different aspects of Arctos. We're going to search now on publications and projects, and we'll kind of linger here a little bit. So now we're going to search on a marmot, I guess.
You have no choice.
And what we see here in the results are all the projects within Arctos that mentions marmots, and we also have all the publications that involve marmots. Some of these will have actual cited specimens. Some of them will actually have attached media, so you can go directly and download the publication. Some of them will just be links to, online versions of that particular publication. When we're nagging you about publications, this is where one of the places, many places it ends up. So let's take a look at this particular project right here. Okay, so this is a project page. So we're going to spend a little bit of time talking about project pages. So project pages are essentially aggregation sites. These are stable URLs that we generate whenever we have some kind of specimen transaction. So this will include loans for specimens based on a research question or topic. It may be an expedition, new accessions coming in based on, let's say, the Guatemala project. So those would all generate new projects that we enter in. We identify, key participants. We will include a description, a brief description. This can come from you directly, or we will nag you for it, or we can grab it from the abstract of, let's say, a publication. If you scroll down, these aggregation sites will actually, as we start entering information that's relevant to that particular project or research topic, or whatever activity happens to be, we can actually, these links become dynamically live, and they will be refreshed with whatever the latest information we're adding in. So publications, this is here we're showing that particular project generated, this phylogeographic study actually generated a publication. It cited a variety of specimens. These are the specific specimens used from, in this case, 3 different collections. And again, this kind of shows the crosswalk between the different collections and how research questions are often not constrained to only one particular museum. All these links will open up maps for those particular specimens. And then we have some other information here, other projects that contributed to that specimen list, and then projects that also use the same specimens, so we can start seeing how specimens are actually being used for a variety of different projects. So you can see there's stuff on hantavirus and Hawaiian things.
Specimens
Do
used.
you want to take a look at that? Let's
Just to show.
Yeah, so this generates the specimen results page, and you can see, which ones are from which collection, and if you scroll down, you can see all of the Researcher-Reptiles. And this will go on, actually, there's a default setting here, I think, of only showing like the first 20, but you can actually set this, which is actually one of the benefits of registering. You can set all sorts of personal preferences, and it'll save it every time you log in. Anything else that we're going to?
No.
Okay. So so let's go back and do a, let's go back and do the OCR search, because I think that's a real thing. So it's under, yeah, there we go.
Research.
No, sorry, that's one. Biological individual here. And so one of the other projects that has been happening with one of my Arctos partners is a huge digitization effort. So the UAM, I think it was last summer, they digitized and scanned herbarium sheets, over 120,000 sheets, I think, in one summer. I mean, just a huge amount. It's still ongoing. But one of the things they were able to do as well, because we can open up one of the images. Go ahead. Even though they massively scanned them, but what they haven't had time to do is actually do a lot of detailed data entry. But luckily, if we open up one of these scanned images here, you can see all the images that relate to that particular one. Oops, that's not what we want to do. Then there's actually a little, tag here. Only part of that is actually handwritten. The rest of it's actually typed up. And so they were able to do some OCR, optical character recognition scanning, and inserted that directly into the database. And so each one was assigned a catalog number. Well, they already have their catalog number, but they were batch loaded up with their catalog number and the media links into Arctos. This allowed then at least quick searches for some keywords. In this case, the locality information was typed, everything else was handwritten, so that's why we have a lot of googling here. But this will allow people now to kind of retrospectively go back within Arctos and do data capture, because they'll be able to look at the scanned specimen sheet as well as the OCR results and fix that dynamically. So, and like I said, we're still able to do immediate searches on at least what was recognized in the OCR, and did a pretty good job on most of the typed portions of the tag. Okay, so now we're going to show some of the external biodiversity resource links. So let's go back to our, I think you log in now, right?
Yeah.
Okay, so go to a process. Carol's going to do a log in here. So some of this, most of the stuff anyone should be able to do. There's no special permissions going on. But we'll log in so we can see some a little bit more detailed stuff.
What's the reason for the login?
The login, well, first of all, Carol's saved preferences, so she'll be able to, more quickly get to what we want to do.
So you can see all my windows are expanded because I use a lot of all the windows all the time. And then you'll see when I do a search right now,
it also defaults to MVZ.
See, I have like 1 million fields because I look at everything, so I like to view all of them all the time. So.
Right. So we're just showing you
so
a little bit of the
preserving customization.
Exactly. We're just showing you a little bit about that. I'll let you know when we get to a section that's maybe permission only. That'll be at the very end. But I wanted to show most of what's capable, for
you actually
anybody.
also need to log in and create accounts if you want to download data.
Oh yes, that's true.
Because
we track that.
We track that.
We track usage. I mean, not in a...
They're like Google Analytics kind of stuff.
Yeah, not in a scary way.
And then, all the curatorial permissions and everything.
So that you can market specimens to us later.
Pardon? Consent?
Yeah,
You
no.
can market specimens to us.
Yeah, right. Yes, it's all for marketing. Though actually, someone, it's Mike here, I don't know, he brought something else up. Yeah, there you go. So I'll point that annotation out because that's actually, that we do have a variety of features within Arctos that allows feedback directly to the Arctos curatorial staff and programmers if there's broken features, or if we also get remarks about misplaced references or misidentified specimens, or questions, like, is there something odd with this? Okay, so Carol did a search on a bird that you can see in this particular, here's an example of the difference between public users and a curatorial user. She has a lot more tabs up here. but we picked this one because we wanted to point out a couple of things. First of all, it's got a lot more identifiers. So this GenBank sequence, yeah, that link will automatically take you to its GenBank entry. And you can see here, these links out will link you back to the MVZ Arctos specimen detail page. It takes you straight back to this page. So let's go back to GenBank. I don't know, do you want to have it say a word, or do you want me to?
So there was something else on here, but we can come back to that.
Yeah, I'll talk about Digimorph in a second. Yeah, in terms of GenBank. So, okay, so a lot of these links are actually dynamic because you don't want us spending time tracking down your specimens and figuring out which ones are entered into GenBank and need to be linked directly back to specimen detail page. That actually only happens if in your initial entry as the author, you format the specimen correctly. So this is basically the typical format, and we actually have, I think, a template worked out to help people who contribute to GenBank to properly link these, so these happen dynamically.
Yeah, basically the format, so you enter in the specimen voucher field, the format would be the institution, so say MVZ, it's the GWID identifier, so it's what you see at the top of the specimen search page, if you want to go back to one of those. So MVZ, bird, the catalog number. So the middle part is the collection code, so the NAM for mammal, but it's always that 3-part identifier at the top of the specimen detail page. That's the format. So if you put that format into specimen voucher field in GenBank, then the dynamic linkages will occur. Otherwise, you have to do it manually, which is not so much.
Is it herp or herps?
Herp, 4 letters, yeah. 4 letters.
So it's the collection code field,
Yeah.
So, but it'll be the same for every herp specimen. So if you just get the format for a herp, it's going to be the same for every.
Right.
Just the only thing that's going to change is the catalog number.
Yeah, and this allows your work to be, replicable. I mean, this is, the scientific method. So if you scroll down, I mean, now people know exactly which specimen you looked at and which, G's and A's and T's and C's you were contemplating. Okay.
And we do also, one more thing, we do have bulk loader templates. So if people are submitting to GenBank, we can provide bulk loader templates, which is like 5 columns in them, and you just give us the data, and we can use it also.
Okay, so let's go back to that. Okay, so this specimen was also chosen because it happens to be really well used. So if you scroll down a little bit more, we also have another external database link, and this goes out to Digimorph, which are high-resolution CT scans of various species and specimens. And you can see here that now you can, I don't know if folks are users of Digimorph, but this is a great resource if you want to look at all sorts of animations and slices and 3D analysis of various species, close-ups, yes. And then you can see here, there's a link back to the MVZ calendar. So.
And you mentioned that was a CT scan.
Yes. Yeah. They had borrowed our specimens specifically for, I'm sorry, what's that?
This is Tim Rowe.
Yes, yes. Yeah, they borrowed those MVZ specimens specifically for their species range. They were looking for sort of taxonomic breadth. I mean, I'm not sure if there's anything specific about this specimen, but it was really because it's a representative of that species. So, okay, so
the project.
Yeah, we can.
You can see the loan list.
Yeah, the loan list. I think that's what we. Are you looking at best?
I'm looking at you, but it's the same.
Okay.
Okay, I'm getting too far now. It's nothing.
Have I strayed off course? I've already strayed off course. Sorry. And if actually there's one, you're not authorized here. Go back to there we go. So scroll up to the top here. I'm going to show one more thing here. I think under this table right here. I think don't we have some barcodes?
No. Oh yeah, we do.
Yeah, we do have some barcodes right here. So yeah, one last thing. This specimen, like I said, has been well used. It also has a tissue sample for it down in our frozen tissue collection, and that's its barcode number. So actually now we know exactly where it is in these giant liquid nitrogen tanks right now. So this, like I said, this specimen has been used in a variety of ways and has a lot of information out available to researchers. Okay, so we're going to show you one last thing. This is, oh, actually, no, that's not true. I'm sorry. We're showing you a couple of last things that are very newly developed, and some of them are pretty much still in beta, and we're actually seeking grant funding to develop them further. So we're going to show you, under search, you can see that we actually have this sort of documents data. We're warning people here. And what most of you already know is that we have an extensive field note book library and archives. And there has been several in-house years of funding to digitize these, as well as an NSF grant to have them digitized and placed online. And we're continuing that effort, but more importantly, we're trying to integrate the field notes with the specimens database, so that way they're sort of put into context. And we have found through the Grinnell Resurvey project, for instance, they are invaluable for validating field sites, for extracting observational data, to extracting ecological information and observations, habitat conditions, that sort of thing. So we have just a couple of samples posted right now. This will be a more robust specimen search one day when we get the funding to do that. But we can show you quickly what we envision. And we're also happy to entertain suggestions too. Like I said, it's in development. So what we have here now is a low-resolution scan for the web of our field notes. This is from Joe Grinnell, 1911. This is his Yosemite trip. And it's a replica of the field notebook page. So you can page through it like, in order, as if you were reading it right here in the library. And we're going to go to page 9 because I know there's some specimens there. And we've also developed this graphical referencing tool, this bounding box tagging, I think we're calling it now. And you can see that there's these boxes here now on the page. And I know it's hard to believe, but if you actually spend a lot of time reading Joe Grinnell's books, you actually are able to actually understand what he's writing. So, so here we have actually field number 664, garter snake. He has some ditto marks here. And what I've done is drawn a bounding box. I've tagged it to the specific specimen over there. We have a bunch of other ones. Here he switches from, there's a couple of snakes to now gophers. So now we have gophers listed over here. If you click on one of these links right there, the first one's fine. We can see the actual specimen right here. And so obviously we don't use the field numbers. Here's the field number right here. The actual catalog number is 2490. Yeah, you can see it's collected by Joseph himself. And if you scroll down here, if I were to encounter this specimen in another searching context, I could also see immediately that it's been tagged in media, which means that if I click this link, it takes me back to the field notebook page, which will allow anybody else to also verify what perhaps we've already verified in terms of locality or what other things. But there's a lot of other information here that's not captured in a normal specimen collection database. Maybe it's important to know what other animals he collected alongside that particular specimen or observed. If, as Carla mentioned, we'll have observational data, and that sort of information is more difficult to glean in a traditional specimen collection database. So this sort of integration we feel is really important, and like I said, we're in the middle of seeking funding for it. So more collective prayers, please. Anything else you want to add about field notes? And then we'll just show you something that happened literally, what was it? What day is today?
Yesterday?
Yesterday. Yes,
In
yesterday.
development. Yeah.
Okay, so like I said, like Carla mentioned, I'm just hopefully emphasizing or letting you believe that this is an ongoing development project. So this just happened yesterday. So actually, I'm going to show you now some curatorial functionality. If you go under Manage Data, go to Location. Let's go to Find Locality. This is the sort of thing that a normal user won't be able to do, but I just want to also show you that as managers of this, and for those of you who also help curate the database, we put in a lot of new tools. So the latest one, I should back up just a little bit. The latest one is a semi-automated georeferencing tool that now is integrated and embedded in Arctos. So that occurs as a sort of a plug-in to the whole Arctos interface itself. It does all sorts of web calls behind the scene to another georeferencing informatics project called Geolocate, which does automatic lookups based on a locality string. So I'm going to demonstrate this, but I also will show you, what it looks like on the curatorial end, because I think that sort of helps clarify a little bit about the locality and coordinate information that you end up downloading from the website. So let's see. We've got a lot of options here. Okay, in specific locality, just put in Bullfrog Lake. Oops. Here's a real look at Jim will recognize this locality. He helped me fix it the first time around. You hit fun. Okay, here we. Okay, so we've got a lot of bullfrog lakes in the
Fresno.
Pardon?
In Fresno.
Yeah, exactly, in Fresno, because there's actually more than one bullfrog lake. So let's hear. Why don't we just click on this one right here? Okay, so this is like I said, most people don't see this part. This is the gory details that me and my students actually see every day. This is the edit locality page. So we have some information about higher geography, which we can change and edit. We have information about localities, which we can change and edit. And we also keep this. I don't think most people are aware, but why don't you keep scrolling down? I'll just show people the lay of the land here. We store all the previous georeferences. So one of the major, not major, but one of the many things that can be corrected in a specimen is the geographic representation of a written locality. We call these determinations, and we realize that since most of these are not supplied by the collector, these are really interpretations. So we actually store different representations. We don't show them all, but we do keep them all. So you can see we've got some here. We scroll to the, okay, well anyway, so here, this is an unaccepted one, or I just depreciated it this morning. Actually, if you show this one's actually from Manus. I know this person actually worked on Manus. She actually didn't have the full scope of information. She just had the raw locality, and that's it. No information about species, era, anything. So hers is like the most naive georeference. Pascal's at the bottom, that was the one I just depreciated this morning. That actually came with a little bit more information. That came from, actually, a correction from Jim Patton about which bullfrog lake. And then I decided to give geolocate a test. So let's just show how I did that, this latest one. So this is a brand new button. Just hit OK. It opens up geolocate. And like I said, it's sending now our locality information, the higher geography, the specific locality, to geolocate. And it actually interpreted that locality as potentially 4 different places. I'm sure what you know what you can do? I just found this out today too. If you hold down the shift key, you can actually create a dragging box and zoom into the. And it, yeah, okay. So it gave us actually 4 different possible localities. If you scroll down at the bottom, we're actually working on a few web services links in terms of like how it displays. It's a little too large right now, but geolocate will rank these according to its internal database and what hits it had in the locality string. So it found some other frog lakes, which obviously are not the ones, but I think they occurred in Fresno County, so we thought, well, it's a possibility. It also found another bullfrog lake, which was confusing to us initially, and actually we had it wrongly georeferenced earlier in a couple of years ago. And then it also actually found the correct bullfrog lake, which is this green one. And so,
sorry, I'm using the wrong mouse. Scroll back out.
Okay, very good. Yeah, so you can actually now just visually georeference things too, because you'll be able to see, spots on the river, rock outcrops, various other features that really just are difficult to do without having on a written locality, especially if this is your own this is your own collection. So here's the green is the one, and actually if you zoom in, actually it takes us to the bullfrog lake a labeled version of Bullfrog Lake.
Will the green one always be the approved one?
Well, that's the one that Geolocate ranks the highest as the most appropriate.
Okay, so
but it's really up to you.
But it's also the right one.
Yeah, but it's it's actually up to you to determine what's the right one. So you can actually, so that's what Carol's going to demonstrate now. So you just click on that green button there. You can actually move that now. So you can take that, just grab it, toss it someplace else because, you know, it's Bullfrog Lake got you in the area, but, you know, I actually know for a fact that particular pike was found above Bullfrog Lake, or, you know, perhaps there's a bullfrog. We actually have no bullfrogs for Bullfrog Lake. So, you know, yeah, the ultimate determination is still manual. But the semi-automatic, that's the semi-automatic part. But the automated part is really just to get you in the neighborhood. And sometimes, perhaps the most conservative approach is best if, let's say, you're working with a foreign locality and you don't have any other information to go on, then you might just accept that, you know, Geolocate's guess is just as good as yours.
So one thing to point out is anybody can use Geolocate, even if you guys can't use the back end part of it from Arctos. You can go to Geolocate's website and they have these same web services there. So you can either batch process things or you can type them in on your own.
Right. Yeah, and then we can also edit the uncertainty. I'm not going to go into uncertainty estimates and things like that. You can attend a georeferencing workshop if you want for that information. Yeah, and then as we adjust that and are happy with it, we can actually push this information back to the Arctos field and save the results. And then you can just go ahead and just close the, there's a little, yeah.
I have a question relating to the project listings. Do you guys enter in projects anytime you get a loan request?
Yes. Yes.
So is that stuff publicly viewable? So if someone, I'm just wondering if people are doing a study where they might not want other people to, you know.
A secret study?
Or be working on specimens and maybe a little bit competitive or something, but.
We have had instances where people didn't want us to put all the information about their studies, so we'll just put a very brief, general title kind of thing, or, you know, not put all the project description in until the person actually is able to publish on it. But we do immediately, because the only way to track that the specimens were all associated with one project, associated with that loan, and then eventually associated with the publication, you assume, that is to make a project. That's the only way to link all those things to each other. So
in addition, like if you were interested in doing a study and you searched on Arctos and saw specimens that you were interested in, you clicked on them and saw that someone else was working on them, you could at least be alerted to the fact that someone else may be doing the same project or.
Right.
Yeah.
I mean,
And sometimes we get loan requests from totally independent researchers for, you know, exhibit the same project, and so it lets us track.
So is that true for in-house loans as well?
yeah.
Any
Yeah.
transaction. Now, if there's particular reasons why certain parts of any transaction, you know, like even things that are permanently stored in Arctos, so let's say the locality of a specimen in a sensitive area, you know, we do have the ability to encumber, what we call encumber certain parts and information from the public record. The curatorial staff will still be able to see it. So those will just show up as being unavailable to the public. And so we have done that in certain cases. And that's possible, so you can talk to folks about. And there could be different levels of that kind of encumbrance of data. Is there any other questions? Sorry.
I wanted to go back to the basic search by the normal person, or maybe sub-normal person. Locality is right up there, and it says any geographic element, but it seems like there's some secret formula as to what it will accept or what order or whatever under that single geographic element.
Well, it's best if you put in a single sort of feature. So if I put in an entire long string of information, you know, that basically just throws up too many search parameters for it to function well. So in that case, I would suggest opening up your options and maybe and then, you know, if it's a particular country or state or province and, put it in the appropriate fields. But if I just want to search, Alameda County, I can just pop that in, and then like a taxa or a collector or Yosemite National Park, then you should be able to do that. We also have this other feature here. If you drop down the geographic feature, you can look on defined, kind of footprints, spatial footprints that maybe are more difficult to capture in a bounding box. So there's a lot of Alaskan stuff in here. But there's Yosemite National Park, for instance, Yellowstone.
Do you need to put the state in there first or last or not at all?
No, you don't have to. If you're looking on geographic search, you don't need to put anything except for that. Just pick out that particular protected area. But if you're in like geographic element, if it's Yosemite and only Mariposa County, then you could do that. But like I said, it might be clearer and faster to actually put Mariposa County here. Sorry, go ahead.
Well, just going to say clarification to Phil. Whatever you put up there in any geographic element or down geographic feature has to be in the locality as it's designated in Arctos. So not all specimens in our collection that are from Yosemite National Park actually have Yosemite National Park as part of their locality description. So you'll miss those.
Yeah.
Okay.
Right.
Except
But if you put Yosemite, so the sort of the less specific.
Right. Except the only difference with that, Jim, and this is brand new, is this Yosemite National Park geographic feature. So what we did was actually take a spatially defined all the specimens. But that is a good point.
I was just using that as an example, but there will be other kinds where that it has to refer to the specific. To an element that's in the locality.
Right.
Or otherwise associated.
The whole locality stream, including hydrology.
Right, yeah, but we got around that, so we started doing that. Yeah, go ahead.
So along those lines, if you're searching based on USGS quad, that would have to be mentioned in the record somewhere?
Yeah, right.
Well, that would be better to do the select and actually just pick it on the map.
Right, yeah, either just pick it on the map, or actually we do have a field for quad information. And that's because Alaska doesn't have counties. Now, we've actually talked about some quadrangle searches and stuff like that, but we haven't actually implemented those. It's a spatial element, like a drop down. Here's my quad, pick the quad visually. But if that's something that's important, we can get to that. Yeah, oh, sorry.
Well, if you just put in a county, there might be half a dozen counties in the same name across the country. And how can it deal with that?
It will pull up everything we have in the database with those
if
counties.
If you use any geographic search, so if you put in Smith County, I know there's like a bunch of Smith counties all over the Midwest, in like 3 different states. So you could do that. I don't know.
So if you only want like California and the county, then put the state.
Then you have the drop down.
Okay, so it's better to use this detailed map rather than just a single element.
I use a single element all the time.
Well,
it depends on the query you're doing.
Yes. Yeah, but for that, I would recommend the fields.
Smith County, California?
No.
Nope.
There you go.
Come on. Oh, sorry, go ahead.
I was going to go back to the field note. So all the ones that we've got online are digitized. Have you guys figured out how to deal with PDFs and making them available?
Yeah.
And accessible through the same kind of specimens, the field note page, kind of links.
If we get the grant, we will.
Yes.
So that's actually one of the things that we've specified. So we'd like to be able to generate them both dynamically for what you've selected. Just drop me down a download link for that particular PDF, or we may just have them stored internally, especially at the project levels. For instance, for some of the Grinnell projects. But yeah, that's a big thing. Yeah.
One other thing that I always worry that we are losing, and what we're going to continue to lose, is where people collect phenotypic data or some other attribute data that ties to specimens, often in table form, Excel files, Access databases, and whatever. We have a lot of it, like morphology, genotypes, etc., etc. What do you think on how we can capture that and connect it to specimen level?
Well, you can upload any. The media basically you can upload Excel files.
Yeah, as long as if it's digital, we can upload it.
Yeah.
So
Can you link it to a specimen number?
Yeah,
Yep.
we can do that too. Yeah.
Yep.
We haven't done that yet.
We haven't done it for Excel files.
You can
although
send me an Excel spreadsheet of your next big project, and we'll try it out.
We've
But
done,
yeah,
yeah.
we could definitely.
I mean, I know MSB is doing that and linking them to sessions, but yeah, you can link any so media can include any format basically, and can be linked to anything in the database, including specimens.
So a lot of people in this room have things going back to Alizon data sets. People looked at phenotypes, measured morphology, microsatellite genotypes, all sorts of stuff. We should start capturing that. This all pertains to the specimens in the